Good evening, everyone. I welcome you all to the 8th live session on NPTEL Bioinformatics Applications and Algorithms course. So, in this course, mostly I'll be discussing about the protein stability, protein folding rates, and how experimental protein stability is calculated. And we'll also be trying to solve some questions on protein stability calculations, effect on mutations on protein stability, as well as how stabilizing residues are identified in a given protein. To start with protein stability refers to the tendency of a protein molecule to maintain its three-dimensional structure under various environmental conditions. This stability is crucial for proper functioning of proteins in biological systems. The stability of a protein structure is influenced by numerous factors, including the sequence of amino acids, interactions with solvent molecules, temperature, pH, and presence of ions and other molecules in the surrounding environment. Understanding protein stability is essential in fields such as structural biology, biochemistry, and drug discovery. Proteins must maintain their native structure to perform their biological functions effectively. Changes in protein stability can lead to protein misfolding, aggregation, and loss of function, which are associated with various diseases, including neurodegenerative disorders like Alzheimer's and Parkinson's diseases. So here I want to talk about the concept Gibbs free energy equation, also known as Gibbs-Helmholtz equation, relates the change in free energy, that is stability, to enthalpy change and entropy change and temperature of a chemical or a physical process. For a protein folding or unfolding process, the Gibbs free energy change can provide insights into stability of the protein structure. So here the equation of Gibbs free energy equation is given as delta G equals to delta H minus T delta S, where delta G is the change in Gibbs free energy, delta H is change in enthalpy of the system, and delta S is change in entropy of the system, and temperature, T is temperature in kelvins. Protein stability is determined by complex interplay of various factors, including protein structure, solvent environment, covalent modifications, chaperone and other proteins, and ligand and substrate. First one is protein structure. The folding of a protein to its native conformation is a result of a delicate balance between attractive and repulsive forces among the amino acid residues. The structure of a protein is stabilized by various interactions such as hydrogen bonds, disulfide bonds, van der Waals forces, and hydrophobic interactions. Next one is the solvent environment. The solvent environment, including pH and temperature, can affect the stability of a protein by altering the balance of electrostatic and hydrophobic interactions among the amino acid residues. Changes in pH or ionic strength can disrupt hydrogen bonding or electrostatic interactions, leading to protein denaturation or unfolding. Third one is covalent modifications. Covalent modifications such as phosphorylation and glycosylation can alter stability of a protein by changing its structure or interaction with other molecules. For example, phosphorylation can change the charge or conformation of a protein, while glycosylation can protect the protein from denaturation or proteolysis. Then we have chaperones or other proteins, which can assist in the folding and stabilization of proteins by providing a protective environment and also promoting correct folding pathways and preventing aggregation and misfolding. Lastly, we have ligands and substrate, which can stabilize or destabilize proteins by altering their conformation or interaction. For example, the binding of a substrate can stabilize the active conformation of an enzyme, where the binding of an inhibitor can destabilize the enzyme. Overall, the stability of a protein is a complex and dynamic property that is crucial for its function and biological activity. Understanding these factors that contribute to protein stability can help in design of more stable proteins for therapeutic applications. So now that we know what factors affect, let's see what are the contributions to the protein stability. First one is hydrophobic effect. So the hydrophobic effect plays the crucial role in stabilizing the native state of protein. When non-polar amino acid residues are buried in hydrophobic core of a protein, they minimize their exposure to the surrounding aqueous environment. This minimization of hydrophobic surface area reduces the unfavorable interactions between the non-polar residues and the surrounding water molecules, thereby contributing to the protein stability. So in this scenario, we understand this hydrophobic effect or hydrophobic collapse is a loss of entropy by protein, but it is gain of entropy from the surrounding water. So this is a driving force for the protein folding. So this burial of hydrophobic residues in protein core residues reduces the entropy as they become more ordered around the non-polar groups. So this entropy decrease is favorable and contributes to the overall stability of the folded protein. Then we have van der Waals forces, including both attractive and repulsive interactions between the atoms, contribute significantly to protein stability. These forces arise from fluctuations in electron density within molecules, leading to the transient dipoles and induced dipoles. In the tightly packed interior of the protein, van der Waals interactions between atoms helps to maintain the structural integrity of the protein's folded state. So these van der Waals interactions are primarily enthalpic in nature. The attractive forces between the atoms in close proximity result in favorable interactions, leading to a decrease in enthalpy and contributing to the stability of the folded protein. Then we have the most important ones, that is hydrogen bonds between the polar amino acid residues. So these hydrogen bonds are mostly observed in secondary structural elements like alpha reses and beta sheets. These bonds contribute to stabilizing the structural motifs and help to maintain overall fold of the protein, fold or structure of the protein. While hydrogen bonds may not be primary driving force for protein folding, they significantly contribute to the stability of native state, particularly in regions where they are involved in stabilizing secondary structures. So even these hydrogen bonds also favor the enthalpic nature by forming stabilizing interactions between polar residues. Then we have the opposing one, that is entropy. The folding of a protein involves decrease in the conformational entropy of the polypeptide chain as it transitions from highly populated denaturation states to a unique fold state. So this decrease in entropy acts as a barrier to folding and opposes stabilization of the native protein structure. Despite this entropy loss, the favorable contributions from the non-covalent interactions outweigh the entropic cost, resulting in a small negative delta G favoring protein folding. So understanding this interplay between these factors is essential for elucidating the thermodynamics of protein folding and stability. Experimental studies, including the analysis of wild type and mutant proteins, provide valuable insights into their relative contributions of each factor to the overall stability of proteins. By investigating the thermodynamics of small molecule interactions and behavior of proteins under denaturation conditions, one can further dissect the mechanisms underlying the protein folding and stability. So here I have a question. So residues with what level of hydrophobicity, whether high, low or medium, are more likely to be buried in the protein core and interact with the what residues, contributing to the overall stability of the protein structure. Consider this in case of globular proteins. So the first question is, residues with what level of hydrophobicity, whether high, low or medium, are more likely to be buried in the protein core, that is, are more likely to have a collapse, hydrophobic collapse, and interact with what type of residues, contributing to the overall stability of the protein structure. Okay, so residues with high hydrophobicity are more likely to be buried in the protein core. This is called hydrophobic collapse, and in the protein core they interact with other what type of residues contributing to the overall protein stability of the protein structure. So it is interact with other hydrophobic residues contributing to the overall stability of the protein structure. So here I have a similar question that is framed. Residues that are what level conserved are more likely to be involved in stabilizing the protein structure and can be predicted as stabilizing residues, whether highly conserved residues, low or medium conserved residues. So residues that are highly conserved are more likely to be involved in the stabilizing protein structure. So this we will see in the next coming slides how. So okay, to overall summarize this, in globular proteins, residues with high hydrophobicity are more likely to be buried in the protein core and interact with the other hydrophobic residues. These interactions help stabilize the protein's folded structure through hydrophobic effect, van der Waals forces, and other non-covalent interactions. This hydrophobic core is essential for maintaining the structural integrity of the protein and preventing exposure of these hydrophobic residues to the surrounding aqueous water. In contrast, membrane proteins are typically embedded between the lipid bilayer, where hydrophobic interactions with lipid tails are crucial for their stability and function. So while hydrophobic residues may still play an important role in membrane protein structure and their arrangement, interactions differ slightly from those in globular proteins. This is due to the distinct environment conditions in which these proteins reside. Therefore, the question is more relevant to understanding the structure and stability of globular proteins. Yeah, then we have STRIDE, which stands for Stabilizing Residue Identification. This server is designed to identify residues within protein structures that are critical for stabilizing the overall protein fold. The identification process involves several criteria, including surrounding hydrophobicity, long range order, involvement of stabilizing centers, and conservation scores. Okay. First factor is surrounding hydrophobicity. As we know, as we just discussed, hydrophobic interactions play a critical role in stabilizing protein structures. The surrounding hydrophobicity of a residue is determined by calculating the sum of hydrophobic indices of residues which are nearby within a specified distance from the central residue. So hydrophobic indices for different amino acids types are obtained from experimental thermodynamic transfer experiments and are listed. Many are listed in A-index database. Then we have long range order, which measures the extent of long range contacts a residue makes within the protein structure. It quantifies the cooperative interactions between the residues that contribute to the overall stability of the protein. The long range order of a residue is calculated by counting the number of residues within a certain distance and normalizing it by the total residues in the protein. So one can calculate a long range order surrounding hydrophobicity using the PDB param server that we have discussed in the last class. Then we have stabilizing center. So these are the regions within the protein structure where long range contacts occur and supporting residues are present in adjacent tetrapeptides. So residues involved in stabilizing centers are identified based on the contact map of a protein, where residues are considered to be contact if the distance between their heavy atoms is less than the sum of the van der Waals radius plus 1 angstrom. Then we have conservation scores. which assess the evolutionary conservation of residues across different protein sequences. Highly conserved residues are more likely to play critical roles in protein structure and function. Conservation scores are obtained by comparing the sequence of the proteins of interest with sequence of SwissProt database using a homology-based alignment approach. To identify stabilizing residues, a consensus approach has been identified. So it has four conditions. So a residue is classified as stabilizing if it meets a specific threshold values for surrounding hydrophobicity, long range order, involvement in stabilizing centers, and conservation score. So these values, what I show here, that is surrounding hydrophobicity, city energy value more than 20, long range order more than 0.02, stabilizing center more than 1, and conservation score of more than 6. This consensus approach identifies a residue as stabilizing. So this trade server combines these criteria to identify stabilizing residues within a protein structures. Users can input protein structure data either by providing a PDB code or uploading a file directly. So the server calculates the values for hydrophobicity, long range order, stabilizing center, and also retrieves conservation scores from sequence alignment. It then identifies stabilizing residues based on the predefined threshold values for each criterion. The server outputs a list of identified stabilizing residues along with their corresponding values for surrounding hydrophobicity. So the results are sent to a user via email due to the time consuming nature of calculating conservation scores. So this server aims to provide uh, sorry is a useful tool for researchers studying protein stability, protein engineering, and homology modeling. It offers flexibility for users to adjust threshold values according to their specific requirements. Further, these stabilizing residues in proteins can be classified into different categories based on their location, function, and protein structure. The preferred stabilizing residues and their locations depends on specific protein and its structural features. So here I have listed some examples of stabilizing residues and their locations. So first is core residues. So these are highly hydrophobic residues located in protein interior where they form a tightly packed core that stabilizes the protein structure. Core residues are often highly conserved and can be predicted using methods such as surrounding hydrophobicity approach. Next we have surface residues. These are charged or polar residues located on the surface of the protein where they participate in electrostatic or hydrogen bonding interactions that stabilize the protein structure. Surface residues can be predicted using methods such as electrostatic potential analysis or conservation analysis of surface residues. Then we have interface residues. These are the residues located in the interface between protein subunits or between the protein and its other interacting partners, where they form interactions that stabilize the complex. Interface residues can be predicted using methods such as contact prediction or interface conservation analysis. Then we have disulfide bonds, which are covalent bonds formed between two cysteine residues, which stabilize the protein structure by covalently linking two parts of the protein. Disulfide bonds can be predicted using methods such as the conservation analysis of cysteine residues or the prediction of disulfide bonding patterns. In general, stabilizing residues are often located in regions of the protein that are important for its function or stability, such as active sites, binding sites, or structural motifs. The specific locations of stabilizing residues can vary depending on the protein and its structural features, and different methods can be used to predict them. So preferred, one second. Preferred stabilizing residues are amino acid residues that are commonly found in protein cores or interfaces and are important for maintaining the stability and structure of a protein. These residues are typically hydrophobic and have high propensity to form stabilizing interactions with other residues. The location of preferred stabilizing residues depends on the structure and function of a protein. In globular proteins, preferred stabilizing residues are typically located in protein core, where they form hydrophobic interactions with other hydrophobic residues and stabilize the protein's compact structure. These residues are often buried and inaccessible to solvent molecules. In contrast, surface exposed residues are often hydrophilic and charged, allowing them to interact with water molecules and participate in protein-protein interactions. In membrane proteins, preferred stabilizing residues are often located in the transmembrane region, where they interact with lipid molecules and other hydrophobic residues to stabilize the protein structure. These residues are typically hydrophobic and have a high propensity to form alpha helices or beta sheets. So in protein-protein interfaces, preferred stabilizing residues are often located in the interface between two proteins and form specific interactions with the residues on the other protein. These residues are typically surface exposed and can be either hydrophobic or polar depending upon the nature of the interface. The identification of the preferred stabilizing residues is important for understanding the structure and function of a protein and for designing the stable and functional proteins. Computational methods such as molecular dynamic simulations, energy minimization, and structural analysis can be used to identify the preferred stabilizing residues and predict their locations in protein structure. Experimental methods such as X-rays, crystallography, NMR can also be used to identify and validate the locations of preferred stabilizing residues in a protein, but they are still time taking. So here I wanted to start with the concept of mutations. So mutations are changes in a DNA or protein sequence. They can arise spontaneously from errors during the DNA replication, mitosis or meiosis, or from damage to the DNA structure, or from mutagens, which are physical or chemical agents that can cause genetic changes. Mutations can result in several changes to a DNA structure, such as substitution of nucleotides or the addition or removal of nucleotides. There are several different types of DNA mutations that are important to understand. Point mutations. One type of mutation is known as a point mutation. Point mutations are mutations in which one nucleotide is replaced by another. Some specific diseases caused by point mutations are certain cancers, neurofibromatosis, sickle cell anemia, Taxas disease, and even color blindness. There are three types of point mutation, that is silent, missense, and nonsense. Another type let's start with the missense, which is a mutation that results in a different amino acid sequence or a primary structure in the product protein. The impact of missense mutation can vary. In some cases, the missense mutation result in amino acids that are very similar to each other. So in these scenarios, these types of missense mutations are known as conservative missense mutation. For example, if a nucleotide that codes for lysine is mutated and now codes for arginine, this will not have a significant impact on the protein because both lysine and arginine are basic amino acids. However, if a missense mutation changes the hydrophobic amino acid such as phenylalanine to an acidic amino acid such as aspartic acid, then that could have a very significant impact on the function of the protein. Then the last type of point mutations is nonsense. The last type is a nonsense. A nonsense mutation is a mutation that results in the formation of a stop codon. There are only three possible stop codons. If one forms through mutation, it can have a tremendous impact on the protein. For example, if the nonsense mutation happens at the beginning or middle of a protein, this can result in a significantly truncated protein that may not function at all. If the mutation happens at the end of a protein sequence, the effect may not be as effective or significant. So here I have the 11th amino acid of a protein X should be serine. However, due to mutation, now it has changed to alanine in that position. So what type of mutation could have this caused? So serine is changed to alanine. Yes, please. So the first one is missense mutation, which is a mutation that results in a different amino acid sequence or primary structure in the product. So the second question is, which type of mutation results in an incomplete polypeptide because of an early stop signal? Yeah. So it is a nonsense mutation. A nonsense mutation is a mutation that results in the formation of a stop codon. So here in protein stability, as we discussed, delta G represents the difference in free energy between the folded and unfolded state of a protein. To calculate change in stability upon mutation, we use the following, that is delta delta G of mutant minus delta delta G of wild type. So sometimes even this delta delta G formula is given as delta delta G of wild type minus delta delta G of mutant. So either ways it depends upon the what sign and what, whether we are considering delta G as Gibbs free energy of unfolding or delta G as Gibbs free energy of folding. So the delta G formula depends on that. Overall, whatever I said, the value delta delta G quantifies the difference in stability between the mutant and wild type proteins, providing insights into the impact of mutation on protein stability. A stabilizing mutation would lead to decrease in delta G, indicating increased stability, while a destabilizing mutation would result in an increase in delta G, indicating decreased stability. Experimental techniques such as calorimetry or spectroscopy can directly measure delta delta G values for specific mutations. In practice, the estimating delta delta G directly from experimental data can be challenging. So, various computational methods have been used in these scenarios to predict the change in stability caused by a mutation. These methods often incorporate features from protein sequence, structure, and energy based calculations to estimate the impact of mutations on protein stability. So there are several databases that are available that has the experimental stability data. So before moving on to these databases, let's get introduced to what is a database. Imagine you have a large collection of different toys scattered all over your room. It is a bit chaotic, right? Now, what if you could neatly organize them into different boxes based on their type, So this is what a kind of database is, that is orderly arrangement of items or information. So to define a database, it is like a super organized filling cabinet for information. It stores a lot of different types of information in a structured way, making it easy to find what you need and when you need. So we have different types of databases, that is relational databases, graph databases, document databases, NoSQL. So previously in our initial classes we have introduction to primary, secondary and derived databases. So here we have relational databases. These databases organize the information into tables with rows and columns. So it is like our big Excel sheet. These relational databases are great when you need to link different pieces of information together, like connecting a customer to their orders. So but in biological databases, scientists all around the world can share what they have learned about different organisms. So if a scientist in Japan discovers something amazing about a rare butterfly, someone in Brazil can learn from it too. Imagine you are a scientist trying to find a cure for a disease. Biological databases are treasure troves for information that can help you to understand how disease works, what treatments have been tried before, and what worked best. For animals and plants facing threats like habitat loss or climate change, biological databases can be a lifeline. By keeping a track of where different species live and how many there are, scientists can figure out how best ways to protect them. In education, students and teachers can use biological databases to learn more about the natural world. It's like having a huge encyclopedia which is full of facts about the animal, plants, and everything in between, right at your fingertips. Okay, here I have mentioned some databases. So these databases often include experimentally measured thermodynamic parameters along with their associated sequence and structural information. So here I have listed some databases such as ProthamDB, ThermomDB, Protabank, and NPTEL. So ProthamDB, one of these, the primary resource, and it contains more than 32,000 experimentally measured thermodynamic data in proteins. It contains experimentally determined thermodynamic data for protein mutants, including Gibbs energy, enthalpy, and entropy changes. ThermomDB is a kind of similar database that focuses on thermodynamic data of protein mutants. And we have Protabank. Protabank is an online repository that stores thermodynamic data and kinetic data for protein folding and kinetic stability. MPTherm is a like-to-like match for Protham, but this is a specific database for membrane proteins and their mutants. So it contains only thermodynamic information related to membrane proteins and its mutant. So it currently has around more than 7,000 mutations from over 300 membrane proteins. So this could be effective resource for understanding the stability of membrane proteins and also differentiating how this stability differs from others. So here I wanted to discuss about Protham mainly, which is because it's one of the vast collection. So Protham offers a vast collection of thermodynamic data related to protein stability, including parameters such as delta G, transition temperature, Tm, heat capacity change, and many more. So this comprehensive data set enables researchers to analyze the thermodynamic properties of proteins under various experimental conditions. In addition to thermodynamic data, Protham provides structural information such as PDB codes and mutant structure information as well. It also has sequence information including protein information resource and Swissprot Uniprot IDs. It also has detailed information on experimental conditions including temperature, pH, buffers, ions, and additives. And it also has information about experimental methods such as circular dichroism, differential scanning calorimetry, fluorescence spectrometry, NMR, and offers insights into techniques used for data acquisition. As I mentioned previously, ProthamDB also includes data on protein mutants, allowing researchers to study the effects of amino acid substitutions on protein stability. Protham has an search and retrieval option. Just now, in some time, we'll go. And it also has visualization tools. The database provides visualization tools for displaying the protein mutants and the surrounding residues, aiding in the analysis of protein structure relationships. Users can visualize the mutations within the protein structure and explore the spatial arrangement of surrounding residues to gain insights into protein stability. So overall, Prothom serves as a valuable resource for studying protein stability, mutants and their thermodynamic properties. By providing access to extensive data and analysis tools, Prothom facilitates research in protein biochemistry, structural biology and biophysics, contributing to advancements in our understanding of protein functions and so here before moving on to this, let's move on and see how Prothom works. So in Google one can type Prothom DB, Prothom database, protein thermodynamic database. Enter the first link from Indian Institute of Technology Madras. So this is the home page for Prothom DB. The home page has given options for home, browse, statistics, tutorial, upload new data, related resources, downloads and other contact and site information. So if we click on a browse information, we have four types of search options that are available. That is simple search, organism based data, cell line based data and newly added DMS data. Okay, now I just wanted to show the simple search option. So in this search options, we have two main panels to search. One is what to search and what to display, because Prothom DB has huge amount of data that is involved as a tables. It also given options to users to decide what columns they want to visit and what columns they doesn't want. So here, okay, let me enter any ID 1234 and see the information. I want to see all the information that is available for the entry 1234. Search. Yeah. So here the results are shown. So entry number 1234, protein name is lysozyme, Uniprot ID is P00698, and it also given its PubMed. So the experimental literature of what. So if we hover our cursor on Uniprot ID and click, the page automatically gets redirected to the Uniprot page, and other information regarding the names and taxonomy, subcellular localization, phenotypes and variants, expression, structure and family and domain information could be obtained. Similarly, if you want to know the thermodynamic experiment behind it, one can select on the DI of this. Okay. Followed by that we have source. So what organism? So this lysozyme protein is from chicken, that is Gallus gallus. Length is 147. PDB ID is 4LYZ. So we can click again on 4LYZ. It gets redirected to the PDB database. And see, this is related to a wild type. So this row in the given Prothom database is corresponds to a wild type, and followed by that we have experimental information such as temperature, pH, buffer name, buffer concentration, ion concentrations, protein concentration, measurement method. So these thermodynamic measures were obtained from differential scanning calorimetry, and the method we call thermal denaturation method, and we have values such as Tm, melting temperature of a protein is 56.38, and we have other parameters such as delta H, delta VH and other experimental information along with the related entries have been mentioned. So this is how the searching in a database does. So if one specifically wants to look for mutant information, let me select single mutant. So the mutation format is here given by PDB ID underscore chain ID colon wild type residue H residue location 95. So this means that H at 94 position is mutated to Y, and the corresponding change in stability values is given as delta delta G or sometimes delta Tm based on the type of experiment, whether it's a chemical denaturation experiment or a thermal denaturation experiment. So this is how a relational database, so it is a relational database that is available. So detailed tutorial has been provided along with the explanation of the terms, that is NO stands for entry number, protein name, EC number, other things. So here next is we have sources of data for biological thermodynamic databases. So this can vary, but some common sources include experimental measures, computational calculations, and literature reviews. Thermodynamic databases often include data from experiments that measure the thermodynamic properties of biochemical molecules such as protein, nucleic acids, and metabolites. These experiments may use techniques such as calorimetry, spectroscopy, and chromatography to measure enthalpy, entropy, and Gibbs free energy of the molecules. So computational calculations means mostly calculation of statistical potentials or using the force field based calculations. So these thermodynamic databases may also include from computational simulation and calculation, such as MD simulation or quantum mechanics calculations. These calculations can be used to predict the thermodynamic properties of molecules based on their structure and composition. Some thermodynamic, then we also have from literature reviews. Some thermodynamic databases include data that has been curated from scientific literature, including both experimental and computational data. The curators of the database may review the literature and select the data that meets certain criteria such as accuracy of the measurements and reliability of the methods used. Then we have data sharing. Some researchers and organizations may share their thermodynamic data with databases as their way to contribute to the scientific community and promote further research. This can include data from their own experiments or calculations, as well as data that has been compiled from other sources. Overall, the data in biological thermodynamic databases is typically collected from a variety of sources and is carefully curated and validated to ensure its accuracy. So mutations can affect protein stability in several ways. Proteins are complex structures that fold into 3D shapes to carry out their biological functions. Mutations can disrupt the delicate balance of interactions between the amino acid residues in the protein, leading to the changes in protein stability, folding, and function. Here are some ways which proteins can affect the stability. That is, we have destabilizing mutations. Mutations that remove or introduce amino acids with unfavorable properties, such as charged or bulky residues, can destabilize the protein structure. Destabilizing mutations can disrupt hydrogen bonds, salt bridges, hydrophobic interactions, leading to unfolding of a protein. For example, a glycine to aspartate mutation in thermophilic enzyme citrate synthase destabilizes the protein and reduces its thermal stability. Then we have stabilizing mutations. So mutations that introduce or remove amino acids with favorable properties such as hydrophobic residues or proline can stabilize the protein structure. Stabilizing mutations can enhance hydrophobic interactions and promote folding of a protein. For example, the substitution of glycine with proline in collagen stabilizes the triple helical structure of the protein and prevents its degradation. But while both destabilizing mutations and stabilizing mutations are known to be part of several diseases. So disulfide bonds are covalent bonds that form between two cysteine residues in the protein and stabilizing the protein structure. Mutations that introduce or remove cysteine residues can affect the disulfide bond formation and stability of a protein. For example, The substitution of cysteine with serine in insulin prevents the disulfide bond formation and leads to the decreased protein stability and activity. Finally, mutations that changes the conformation. Mutations can also cause conformational changes in protein structure, leading to altered stability. For example, a point mutation in p53 tumor suppressor protein can lead to a conformational change that disturbs the protein stability and leads to cancer. Overall, the effect of mutations on protein stability depends on their specific location in protein and the resulting changes in interactions between the amino acid residues. Understanding the effect of mutations on protein stability is crucial for predicting the impact of genetic variations on protein function and disease susceptibilities. So with this I am done. So I'll just be moving on to this NPTEL demo. NPTEL database. This is a home. Just give me 1 minute. So this is NPTEL is similar to Proteome, but this is a database that is specific for membrane proteins. So it has search option similar to the search options of Proteome. We have search options in NPTEL as well, that is options to search and columns to display. So here let me take import ID 1C 3W, select all and submit. So it gives information. So this particular entry protein, that is bacteriorhodopsin protein, has thermodynamic data available for wild protein as well as for mutation, that is 62 mutation data is available. So this 62 mutation data has been given using the chemical denaturation studies. So mutation L13A, or according to UniProt numbering L26A, has a change of stability value minus 1.3. So here we see some mutations have negative values while some has positive values. I don't think so. In this case there is a destabilizing mutation. But even these mutations have both stabilizing and destabilizing effects. So this is what the explanation I have planned. Now that I have some questions. First, which of the following factors influence protein stability? Whether it is temperature and pH, whether it is solvent environment, presence of ions. So all of the above. Yeah, so all of these factors of protein stability is influenced by factors such as temperature, pH, solvent environment and presence of ions. Which factor opposes the stabilization of the native protein structure? Whether hydrophobic interactions are against protein folding, hydrogen bonding is against, or chain conformational entropy, or van der Waals interactions. So wait for a minute for others also to go through question and answer. we studied in the initial slides, hydrophobic effect, hydrogen bonds and van der Waals interactions favor protein folding, but while the chain conformational entropy opposes the folding because of loss in entropy that happens with the protein in case of folded protein state. So overall, the balance between the forces promoting folding and opposing folding is the overall protein stability value. So how was stabilizing residues in protein identified computationally? So here, how are stabilizing residues in proteins identified computationally? Yeah, so by analyzing the surrounding hydrophobicity, long range order, stabilizing center and conservation scores, one can identify the stabilizing residues. So this is the phenomena behind the stride server. So given delta H, that is change in enthalpy is minus 100 kilojoule per mole and change in entropy is 150 joule per mole Kelvin and temperature is 298 Kelvin. So calculate the change in Gibbs energy for a protein using the given values. So here I'll give you the formula. So the formula for calculating delta G is minus delta H minus T delta S. given delta H equals to minus 100 kilojoule, delta S equals to 150 joule Kelvin per mole. Okay, everything is per mole. So now I wanted to convert into same units because one is in kilojoule, other is in other is in joules. So here I'll convert 150 kilojoules by 1000 per Kelvin mole. So this will become 0.15 kilojoule Kelvin per mole. So now let me add this into the formula, that is minus 100 minus of 0.15. So this is, we have kilojoule per mole. Okay, I'll write this at the last, into 298. So here we have kilojoule per Kelvin per mole into Kelvin. So Kelvin gets cancelled. Both the units now become now both the units have become kilojoule per mole. So we can directly subtract, that is minus 100 minus 44.7 kilojoule per mole. So it is 0.7. So this is how delta G is calculated. So I'll wait for a minute. Kindly go through the solution. So again, to re-stress the practice on this, I have similar question. If change in enthalpy for a protein unfolding process is minus 80 and the change in entropy is 200 joule mole per Kelvin, calculate the change in Gibbs free energy. So this is similar to what we have solved previously. Try to solve. I'll wait for some time. delta S is 200 joule mole Kelvin. Temperature is 273 K. Again there is a unit mismatch, so let's convert delta S. So 200 by 1000 kilojoules mole Kelvin, which is equals to 0.2 kJ mole per Kelvin. So delta G equals to minus 80 kJ minus of 0.2 kilojoule mole Kelvin into 273 K, so which is approximately minus 34.6 kJ per mole. So this is how calculations are done for calculating change in enthalpy for a protein folding process or an unfolding process based on the entropy change and enthalpy change given. So now I have what does delta G represent in context of protein stability. So it's a combination of enthalpy and entropy which gives us difference in free energy between folded and unfolded states of the protein. So what is the primary purpose of databases such as Protherm, ThermomDB, NPTEL or any other database? So mostly these experimental databases are for information sharing, which provides the experimental data that is available in protein stability and mutants. So which type of mutation results in stop codon formation? have done this already. It's a nonsense mutation which results in the formation of premature stop codon in mRNA sequence. What impact do destabilizing mutations have on protein stability? So what do destabilizing mutations do? Whether these mutations stabilize the protein structure, have no effect on protein stability, promote unfolding of the protein, or they enhance hydrophobic interactions. Yeah, they promote unfolding of the protein. So here I have a question. If the conservation scores of a residues, the score of 1 represents most what residues and a score of 9 represents most of what residues. So whether the score 1 represents conservative or variable residues, or 9 represents conservative or variable represents, or both represents same as variable or conservative. So the lower score represents the variable residues and the higher score represents the conservative residues. So name a web server for identifying the stabilizing center in protein structures. So here most of these servers I have discussed already, but there is one particular program, SCID site. So the question is not about stabilizing residues, it is about the stabilizing center. So SIDE is the one that is used for identifying the stabilizing center. SIDE is a program to identify stabilizing centers from known protein structures. These are the residues involved in cooperative long-range contacts which can be formed between various regions of single polypeptide chain, or they can belong to different peptides or polypeptides in a complex. The server takes a PDB file as an input and the result is presented in a graphical or text format. This program is intended to select the stabilization centers, that is amino acids involved in contacts which are important in maintaining stability of a protein. Two residues are identified as a forming long range contact if they are at least 10 amino acids apart in sequence and they have at least one heavy atom separation smaller than the sum of their van der Waals radius. So that is, structurally they are contacting, sequentially they are separate. That is how a stabilizing center is defined. So here I have a question regarding long range order. A protein has 500 residues and 200 contacts. Residue alanine 45 has contacts with aspartic acid 46, glycine 47, alanine 44, valine 71, isoleucine 60. So what is the long range order of this residue? So here I have added the formula for long range order also. To calculate the long range order, so here in long range order, n of ij is the separation, is a binary number. The separation between the two residues is greater than 12, its value is 1. If not, it is 0. And capital N is the number of residues in protein. Let me move on to my writer. So this is what Nij has been given. So with respect to residue 45, let's calculate absolute value of I minus j, where I and j represents the position of these amino acids. So 45 minus 46 absolute value is 1. 47 minus 45 or 45 minus 47 and its absolute value is 2. With 44 it is 1 again. With 71 it is 16. Oh sorry, it is so with this it is 15. It is 26. So according to our condition, if I minus j is greater than 0, n value is 1. If not, it is 0. So in the first case, so since the separation between two residues is less than 0, here we have n value as 0. Similarly, in the second case, separation is again less than 12, so we have 0. Third is also, and in the last two cases, the separation is more than 12. So here we have 1, 1. So according to the formula, 0 plus 0 plus 0 plus 1 plus 1 divided by 50, that is 2 divided by 50. Is the long range order for the residue alanine 45. Okay, now that we have got here, let's try to calculate long range order for residue valine 71 now. from the same condition I am changing alanine 45 to valine 71, and we'll try to find the contacts. Considering valine 71 has contacts with alanine 45, aspartic acid 46, glycine 47, alanine 44 and isoleucine 60. So what will be the long range order of this residue? Okay, let's move back here. So let's calculate the separation. So consider this as I, and the j values are given in a column here. First we have is 71 minus 45, which is 26. Yes, please.
So is it 40%?
Yeah, we'll check that once we are calculating. But long range order values will never be for percentage. They are like decimal values are given as a float.
Sir, I mean point 0.4.
Okay, point 0.4 is for previous one.
Okay.
So now here what I wanted to do is I'm just changing the uh, this is 0.4 is for alanine 45, 0.04 is for alanine 45. So
Okay.
what if instead of alanine 45 consider valine 71? So that is what we are doing here. So next we have separation here is 25, separation here will be 27, and separation here is 11. So the NIG value according to our phenomena, that is the separation should be more than 12. So we have 1, 1, 1 and 0. So long range order will be, there should be one more, right? 46, 47. So we have one more residue. Okay, we have one more residue 47 also, whose separation is 24 and it says 1. So now long range order is given as 1 plus 1 plus 1, 0 plus 1 divided by 50. It is 4 divided by 50. It is 0.08. Yeah, so the more connected the residue, more is the long range order. So in the previous case, in previous case for for the similar type of residue we have 0.04. Now the number of contacts has been doubled in this residue, so it has 0.08. So this is how a long range order is calculated. Now I have some more questions before we wind up the session. In a protein, if glutamic acid is in the vicinity of lysine, is mutated to valine, what will happen to the stability? So what condition will happen if a lysine is mutated to valine? So if
Sir, option C.
Yes. Wait for others also to answer. So when we see here, one negative charged and the positively charged amino acids are close. When we replace with one of the charged residues with a valine which is a hydrophobic residue, the formation of ion pairs will be removed. That is, a ion pair is removed. So it is D.
Okay, sir.
So when these two are in close, the chances they can form an ion pair. But if I remove any one of them, there is no chance of forming an ion pair. So given the surrounding hydrophobicity of threonine 27, and if it is surrounded by two alanines, one valine, two threonines, two glutamic acids, and one serine, what will be the hydrophobicity? What is the total surrounding hydrophobicity? So the surrounding hydrophobicity is given as sum of the hydrophobicity indexes of the neighboring residues. So we have two alanines, one valine, two threonines, two glutamic acids, and one serine. So accordingly, I have arranged so 2 into 1, which is a hydrophobic index of glycine. So sorry, alanine. Then we have one valine. So accordingly, I have added the hydrophobicity index. So this gives the surrounding hydrophobicity of the given residue. So it is 2 plus 2 minus 2 minus 4 minus 2. So it is the surrounding hydrophobicity is minus 4. So here further I have some more information regarding the databases themselves. Uh, so here given when someone asks you to calculate the value of delta delta G based on given its wild type and mutant, two types of formulas, that is wild minus mutant or mutus minus wild. It's based on the convention, sign convention or the process convention that is used. So in most of the studies, it is used as delta G wild minus mutant. But when it comes to thermodynamic studies, equal weightage has been given to delta G mutant minus delta G wild type. So it's always the sign convention matters. So if your delta G values are negatives, wild minus mutant is to be preferred. If your delta G values are positives, mutant minus wild type should be preferred. Do make a note of this, and that's all for today. If any other
Sir,
questions I can take, yes, please.
sir, is this for amino acid or for nucleic acid?
No, generally stability values have been given for proteins, so it's for amino acid, mutation of amino acid.
Okay, sir. Sir, I had a small question.
Yes,
Actually, I usually attend Sunanda sir's class
okay.
on Fridays, as I'm free on Friday. Uh, Actually, I wanted to ask whether I'm actually a master's student in molecular biology. So I was interested in studying these subjects. So does your lab offer any sort of internship like after completion of this program?
Yeah, regarding this internship, the people who get that gold standard or the what top standard marks, they will be offered an internship through NPTEL and the course instructor Michael Rumia sir. Beyond
Okay.
that, also we have some intern or project opportunities. For those, kindly go through our lab website. Like you can just type PBA lab or Michael Chromia's lab.
Okay, sir.
You can get, and you can just contact sir through the mail. Through
Oh, okay, sir.
NPTEL, only to the top students we offer, like who score
Oh,
that gold standard or diamonds, that standard marks, top standard marks.
sir, like, what are those standard marks? And actually,
Like it's
by mistake,
NPTEL benchmarking. So if you take top 10% of the students, like to
okay.
be precise, I'm not sure what precisely how they calculate, but in my knowledge, it's like if 10 people write top 10%, like one is the who is the top rank. If 100
Oh,
write top 10, like that.
okay. Sir, and also like by mistake, I forgot about one assignment to complete. So will it matter or
Since it's only one assignment, you can do well in other assignments and score good there. Because
okay.
NPTEL has a timely operated backend system, there is no reversion of assignments will happen through NPTEL. Or for more information, you can also contact NPTEL team, because that assignments hosting and closing them is not in our hands. It's in with NPTEL.
Sir, I mean, what amount of what percentage is considered in the final exams? Like, what percentage of assignment marks is considered in the final exams? I mean, in the final score.
Yeah, even those weightage and all, you can just ask in that Q&A session of the score. The instructor will reply you back. We are not
Oh,
allowed to, as a TS, we are not allowed to display those.
okay.
But it's not more than 50% in my knowledge. It's somewhere less than 50% only. One assignment doesn't affect you much in your markings.
Or if we do well in final exams.
Well, and other assignments, if you do well, it won't affect you.
Okay, sir. Sir, and may I know on what field you are working on?
Yeah.
I mean,
Okay.
your PhD is on.